Okay, so this is the video presentation for chapter 24, which is on fungi. Okay, so we are covering this chapter, first of all, to learn about fungi, but also because the next couple of weeks in lab, this week and next week, I believe, we're going to be dealing with fungi. So this week in the lab, you're going to be looking at pictures, which you would have been looking at slides, of different fungi under the microscope, and you're going to be labeling structures, and you're going to be figuring out, we've got 4 unknowns that you're going to be figuring out which division they belong to. So we're going to talk about the 3 divisions that we're going to look at in lab. And then next week in lab, you're going to be hearing about, looking at the data from an experiment where we're actually going to cross 2 mating types, 2 opposing but compatible mating types of a fungus called Sordaria firmicola. And you're going to be looking at the results of that and relating it back to genetics. Okay, so everything that you learned in those first couple of chapters, don't forget about it because we're going to still be applying it. Okay, so we have to have just a basic kind of understanding of the kingdom fungi, what they look like, what the different divisions look like, and how they reproduce, okay, so that we can get through the next couple of weeks in lab. So again, this corresponds to chapter 24 in the OpenStax textbook. And by now, hopefully you've done your vocabulary and you've done your chapter outline. You've got your PowerPoint presentation hopefully in front of you so that you can follow along. And you want to also have those Are You With Me's available. Okay, so let's go ahead and start. So first off, fungi belong to the domain Eukarya, which means that they are eukaryotic or eukaryotes. Okay, so this term right here, hopefully you didn't forget what that means. You should have learned about that in General Biology 1. Eukaryotic organisms have a membrane-bound nucleus and other membrane-bound organelles. Okay, so the genetic material of a fungus, right, is going to be held in a compartment called the nucleus, which will have a membrane around it. Other organelles like mitochondria, the endoplasmic reticulum, the Golgi apparatus, ribosomes, those are all going to be found in fungal cells as well. What we won't find are chloroplasts because fungi are heterotrophic. They're not autotrophs. So they cannot make their own food. They do not possess chloroplasts. Instead, fungi are going to be other eating, which the term heterotrophic literally means other eating. So they cannot make their own food. They rely on other organisms for nutrition. Reproduction we're going to talk about in a little bit more detail, especially when we get into the life cycles of some of these fungi. But some are going to reproduce asexually, and some can reproduce both sexually as well as asexually. And they'll go through, they'll alternate generations or alternate between a haploid and diploid state. However, the most prominent state, most of the fungal life cycle, is going to be spent in the haploid condition, which is the complete opposite of us. So this is going to be interesting when we study genetics. So remember for humans, for eye color, you inherited 2 alleles for that eye color gene, right? And those alleles are going to interact in some way to produce a phenotype. The same thing with the corn that we've been studying in lab. Corn kernel color, right? Corn kernels can be either purple or non-pigmented or clear or yellow, we'll say, okay? So we have 2 alleles, right? They could both be purple, they could both be yellow, or one could be purple, one could be yellow. But that combination is going to give you a phenotype. Well, in fungi, that's not the case. Instead, they only inherit 1 allele for every gene, and that 1 allele gets expressed. Okay, so in lab next week, when we start to look at fungal genetics, we're going to study spore color. And spore color is going to be controlled by actually 2 genes. The first experiment that we do to look at spore color, we're just going to follow 1 gene. And so whatever allele is inherited, that's the color of the fungal spore. Okay, so genetics is pretty easy to study in fungi because they spend most of their life cycle as haploid organisms, not diploid like us. Okay. they play an essential role in pretty much every ecosystem on the planet. They are decomposers, and so they are going to break down dead or decaying organic matter, and they're going to put the nutrients from that matter back into the ecosystem. So they're really, really important in the cycling of nutrients. Oftentimes we will find fungi involved in mutualistic or symbiotic relationships with other organisms. Okay, and if it's mutualistic, that means that both organisms benefit. We're going to go over some of that ecology stuff in just towards the end of the chapter. And we'll look at both of these. So we'll look at mycorrhizae. Mycorrhizae are going to be an association between plant roots and fungal parts underneath the ground, so in the soil. And we'll talk about what that relationship is like. Both benefit. And the second, which is my favorite, lichens. Love lichens. This is going to be a symbiotic, mutualistic relationship between a fungus and either a photosynthetic algae or a photosynthetic cyanobacteria. Okay, either one. Again, we're going to talk more about those in a little while. Some fungi are disease-causing for plants or animals, and some have commercial applications. Okay, so again, this is just kind of the intro. We're going to touch on all of this stuff as we work our way through this chapter. Okay, so to start off, the term mycology, this is the study of fungi. Okay, and if you are a biology major, you may have a chance when you get to your 4-year institution after transferring from us, you may have a chance to take a course in mycology. If you do, I highly suggest it. I took a semester-long course and it was really cool. We're only spending, a chapter in a lab on this kingdom, but you could spend a whole course, right, or multiple courses studying this. So I got a chance in college to take a course called mycology. It was just an introductory course, but it was really neat. I got to learn a lot about the kingdom. And one of our assignments, actually our big lab assignment, was to collect and classify at least 20 different species of fungi, as well as 20 different species of lichen. And that was hard, but it was fun. It got us out in the field. That's when my love of hiking kind of took shape. I hiked through so many different trails in the area. And still to this day, when I'm out on hikes, I just did a 13.5-mile hike a couple weekends ago to Indian Head up in Lake Placid. The one thing that I find myself consistently doing as I'm hiking is looking for fungi and looking for lichens. And my husband, who comes on every hike with me, he knows it. So he points stuff out to me as well. And, he rolls his eyes at me, but it's pretty cool. So anyways, so mycology is the scientific study of fungi. Fungi appeared in the fossil record about 450 million years ago. So they've been on our planet for about that long. And again, they are in their own kingdom. So they're in the domain Eukarya. And then for kingdoms, in the domain Eukarya, we've got animals, plants, fungi, and protists. So fungi are in their own kingdom. They've been shown to have some animal and some plant-like characteristics. However, scientists believe that the fungi are actually probably more closely related to animals than plants, okay, which a lot of times students find hard to believe. So let's talk about some of those characteristics. So here's a plant-like characteristic. If we look at fungal cells, their cells are surrounded by a cell wall. We don't find that structure in animal cells, but we do find it in plants. However, when we take a closer look at fungi have the carbohydrate found in their cell walls is called chitin, okay, where plant cell walls have cellulose. So even though they have a cell wall, it has a totally different structural polysaccharide or carbohydrate when we compare it to plants. And in fact, chitin is the same material that we find in the exoskeletons of organisms that belong to the kingdom Animalia. Okay, so again, more animal-like than plant-like. Okay, so they have thick cell walls and they contain a structural polysaccharide called chitin. And that same structural polysaccharide is found in exoskeletons of some animals. That cell wall is going to be there to protect the fungal cell from desiccation. So this term right here, desiccation, means to dry out. So it prevents water loss. And it also helps to protect these cells from predators. Most fungi, not all, but most are going to be non-modal. Okay, so they're not going to be moving around at all. There is one phylum that does not follow that rule, but most are non-modal. Okay, you can see right here we've got some fungi. We've got a couple of Basidiomycota, and we even have a couple here, Basidiomycota. These are typical mushrooms that you've probably seen. They have a typical cap and stalk. If you flip that cap over, you would find gills. Okay, now this bright coloration here, that's a warning sign. Not all brightly colored mushrooms are poisonous, but I would definitely stay away from those that are. In fact, I wouldn't eat mushrooms unless you knew what you were doing. My mycology teacher also used to cook for us, and he would take us out foraging for edible mushrooms, but I don't trust myself. I can't tell the different species apart well enough to actually do that myself. So again, just be careful. I get my mushrooms from the grocery store. Okay, so growth. Members of the kingdom fungi could either be unicellular or multicellular. Okay, most are multicellular. Unicellular fungi are called yeasts, and you've all heard of yeast, right? You've heard of baker's yeast and brewer's yeast. Here is the scientific name for baker's yeast, Saccharomyces cerevisiae. Okay, and maybe you've unfortunately heard of this yeast as well, Candida. This causes all kinds of different infections. One that you may have heard of is called thrush. So thrush usually will be in the oral cavity. So if you've ever had or known someone who's ever had a thick white layer of material on their tongue and even on the roof of their mouth, that could be thrush caused by this yeast called Candida. Usually people who end up with thrush, they've got some type of a compromised immune system, or they've somehow thrown their normal flora off. Maybe they're on an antibiotic that's killed off good bacteria, and therefore organisms like Candida come in and they kind of take over. Thrush is not dangerous, but it's pretty nasty. Okay, so fungi that are single-celled are called yeasts. Okay, fungi can also be multicellular. Okay, most are multicellular, and they'll alternate between a vegetative state, which means just normal growth, and a reproductive state where they are reproducing. Okay, in the vegetative state, multicellular fungi are going to be composed of a tangled mass of thread-like structures, and each one of those threads is called a hyphal strand, or hypha. Okay. Reproductive state, you get more of a structure. It's not just this tangled mass of thread-like structure is called hyphae. Instead, you get an actual structure. And that structure, what it looks like, depends on the species. Okay. Oftentimes, we'll call the reproductive structure the fruiting body. Okay, let me back up for a second. Okay, and these mushrooms right here, what we're showing you is the fruiting body. So the cap and stalk, again, if we flip the cap over, you'd find gills. It's on those gills where spores are being produced. Okay, so that would be an example of a fruiting body, a reproductive structure. Okay, so again, fungi can be single-celled or multicellular. And if they're multicellular, they usually will alternate between a regular growth, vegetative state, and a reproductive state where they're reproducing. Okay. In the vegetative state, they're going to be composed of these thread-like structures called hyphal strands. The reproductive state, they will form some type of a fruiting body. And again, that structure is going to be different depending on which species of mushroom we're looking at. Species of fungi we're looking at. Okay. When you have a bunch of hyphal strands in a mass, it's going to get a special name called a mycelium. Okay, so that's the whole collection of hyphal strands. That's the mycelium. And a mycelium can grow on the surface. It can grow into the ground. It can grow into soil or into any kind of decaying matter. It can grow into leaf litter, for example. Some fungi can even grow their mycelium in liquid, or if they're parasitic, they can also grow into living tissue. The largest organism on the planet is actually a fungus. It's the honey mushroom, which is called Armillaria solidipes. And this fungus can be found, it's found in eastern Oregon. And it's been found to spread across more than 2,000 acres, and it's found underground. And it's believed to be at least 2,400 years old. That's pretty incredible, okay? So again, largest organism on the planet. It's a collection of hyphal strands creating this mycelium, and it's really big and really old. Okay, here's an example of a mycelium of an organism that happens to be a human pathogen growing on an agar plate, okay? And in a couple of weeks in lab, when we do the Sordaria experiment, the genetic cross experiment with the fungus Sordaria formicola, I'm going to actually show you plates like this. And so what this is here, this is the agar plate, and it's just a plastic, it's a Petri dish. You've probably all seen a Petri dish. It's got a cover, right? If you take that cover off, what's going to be in there is this material called agar. Agar is a gelatinous-like material, so it's kind of like, it's got a little bit more of a structure than Jell-O, but it's kind of jello-y. And you know, if you poke at it, it kind of cracks like Jell-O would. And what's in this agar is just nutrients. So everything that an organism, this fungus in particular, requires to grow. So food, any kind of essential amino acids or essential minerals would be found in that agar. Okay? And in this particular example, the scientists here, they're growing this particular fungus on this agar. So the fungus here, hopefully I can say this name, Neotestudina rosati. That's the name of the fungus here. And so hopefully you know that I'm going to screw it up. Neotestudina is the genus name and rosati is the species name, right? So every organism gets a two-part name, genus and species. Genus is always capitalized, where species is lowercase. Both are italicized or underlined if you're writing it. This particular fungus is a human pathogen. It is going to enter through a cut or a scrape, and it causes something called a subcutaneous infection. We're going to talk about what that means in a couple of slides, but just to give you a little bit of a definition, subcutaneous means within the skin, okay? So it's going to enter through a cut or a scrape, and it's going to cause an infection that's subcutaneous underneath that skin, okay? If you look here, you're not seeing too much because we don't have a microscopic view of it, but if we did, you'd see a bunch of hyphal threads kind of all probably overlapping and crisscrossing over each other. And towards the edges, you see this powdery white stuff? Those are all spore-producing structures, okay? And I'm not sure which phylum this organism belongs to, but those spores might be produced in a structure that looks like this, sporangium. Or there's multiple different structures. We're going to get to those slides in a few seconds. Okay, so this guy is reproducing. It is producing spores for sure. So this whole collection of hyphal strands, again, is called the mycelium. Okay, we don't really use the term, body. We use mycelium. Okay, so let's take a closer look at what these hyphal strands look like. Let me move myself. Okay. So this is really neat. Look at this rotting tomato right here. Okay. Yes, a rotting tomato is neat. So sitting on top and eating that rotting tomato, breaking it down, right, is a fungus. Okay, and if we took a sample of this fungus and threw it on an electron microscope, this is what it would look like. So all of those thread-like structures, those are all hyphal strands. Okay, and in this particular example, it looks like those hyphal strands, like there's separations between the cells. Okay, so some will have that and some don't. So let's take a look at that. So most hyphal strands are divided into separate cells by these dividers called septa. Okay, let's look at this cartoon down here. Let's look at the monokaryotic first. So look at this hyphal strand here. You can see the blue spheres are all nuclei. So you can see that there's one nucleus per separation or per cell. And in between each, do you see the septa, the little divider? And if you look close, you can see a little tiny hole in the septa. So we say that they are perforated. So the cells of a fungus, even though they're separated in this particular example, there's still going to be a connection between neighboring cells, kind of like gap junctions between our cells, okay, between animal cells. So information and materials can flow from one cell to the next, but large things like the nucleus can't. Okay, so any fungi whose hyphal strands are separated by septa, we see that their septa are monokaryotic, which means one nucleus. So there's one nucleus per cell. Now some hyphal strands don't have septa, and so they are what we call coenocytic, okay, no septa between nuclei. And so it's literally just a thread-like filament where there's separate nuclei but no true separate cells. Kind of strange. Okay, you can see that up here too. This would be an example of a monokaryotic hyphal strand or strands. And this is an example of the coencytic condition. Now this last one here, dikaryotic, when some fungi are sexually reproducing, they end up with 2 nuclei per cell. And those nuclei will eventually fuse, but before they fuse, we say that these cells are in the dikaryotic condition, which means 2 nuclei per cell. Okay, so the vegetative state of multicellular fungi is composed of a mycelium, which is composed of hyphal strands, and those hyphal strands could be monokaryotic or coenocytic. And again, if sexual reproduction happens, then we can get the dikaryotic condition. Okay. Where do they live? They can live anywhere. Okay, we find fungi all over the planet, but they thrive in environments that are moist and slightly acidic. They can grow with or without light. They're not photosynthetic, so they don't require it, but they can grow with or without light. Okay, and different species of fungi are going to have different preferences. They also vary in oxygen requirement. Some fungi require oxygen, so they need oxygen to live. We call those fungi obligate aerobes. They need oxygen. Without oxygen, they die. So again, they're called obligate aerobes. The opposite of that are obligate anaerobes. So those are fungi that require anaerobic conditions, which means conditions where oxygen is not present. If oxygen is present, they cannot survive. And the third here are facultative anaerobes. These are organisms that can grow in the presence or the absence of oxygen. Okay, they may function better in the presence of oxygen, but they can still survive in the absence. Okay, so we've got obligate aerobes need oxygen, obligate anaerobes need oxygen poor or no oxygen conditions, and lastly, facultative anaerobes can grow in either oxygen or oxygen with or without oxygen. Okay, nutrition. So we already said that fungi cannot make their own food. They are not autotrophic, they're heterotrophic. They do not have chloroplasts. Okay, so they're going to rely on other things to eat. So they are heterotrophic. And the other cool thing is that they exhibit what's called external digestion. So a fungus will infiltrate, grow into, or on top of whatever it's going to eat, right? And it's going to then secrete out of its cells, out of its mycelium or hyphal strands, digestive enzymes. Those enzymes end up on the food, whatever they're growing on or in. That food will get digested by those enzymes outside of the fungal body. And then that pre-digested, nutrient soup pretty much will then get absorbed into the fungal cells. Okay, so they're heterotrophic and they exhibit external digestion. Most fungi are going to be saprophytic. So you can see that word here. They're saprophytes or saprophytic, which means that they, again, are going to feed on dead or decaying organic matter. Some are parasitic, and they can be parasites of either plants or animals. We'll go back to some of that after too. Reproduction. So again, some fungi will reproduce only asexually, and some will produce both sexually as well as asexually. We use the term perfect fungi, to describe those fungi that display both. Okay, they can both sexually as well as asexually reproduce. The imperfect fungi will only reproduce asexually. Okay, and that asexual reproduction is pretty much just mitosis. Okay, we'll look at that too. Whether it's sexual or asexual reproduction in fungi, the end product of those events are going to be spores. Okay, so spores are going to be either the product of sexual or asexual reproduction for most fungi. And those spores are the perfect way for these organisms to disseminate themselves, to kind of disperse or spread themselves. Okay, those fungal spores can get picked up by wind, they can get picked up by water, or they can get picked up by other organisms and brought to other places where, if conditions are good, those spores will then start to grow. Okay, I don't know if you've ever been walking and ever kicked a puffball. Okay, if you've ever done that, you may have seen a cloud of what looks like smoke. But those are all spores coming out. Look at this guy. How neat is that? This little cloud here that's being produced, those are all spores. This structure right here, as well as this puffball right here, that's the fruiting body, right? Usually the product of sexual reproduction, which is going to produce those spores. Pretty neat. Okay, so some fungi can only reproduce asexually, and we're going to be getting into evolution pretty soon in lecture. So I'll just kind of tell you that, and you probably know this already from our meiosis work, that asexual reproduction doesn't increase genetic diversity, where sexual reproduction does. And so from an evolutionary standpoint, those organisms that are capable of reproducing sexually are really more evolutionarily fit, right? It's better because it increases genetic variation in a population, okay, which increases the chances of survival for the species. Okay, however, some organisms just don't reproduce sexually. So let's look at asexual reproduction in fungi. So asexual reproduction can take the form of a couple of different ways. So first of all, mitosis really would produce spores, or we could have budding, and that's what's being shown here in this figure, or something called fragmentation. So let's touch on fragmentation and then budding on this slide. So fragmentation, this is when a piece, usually of a mycelium, so a piece of the body of the fungus gets broken off, for some reason and ends up somewhere else, and then it could still grow. So we've taken a fragment off of the main body, and it's been placed somewhere else, and it's capable of growth. Maybe a hyphal strand, gets broken off, and wherever it gets broken off to, it continues to grow and divide. Okay, that's fragmentation. Budding is shown here. And so in budding, what happens is we have one cell, right? And that cell pretty much undergoes mitosis, except for usually this little bud here that comes off, it splits. is usually smaller. So you end up with two genetically identical cells, but one cell is usually smaller than the other. Okay, so what happens here is you get DNA replication, and then you get the division of the nucleus, and then kind of divvying up of a lot of cytoplasmic components, and then you end up in the end with two cells. Okay. Okay. Let's look at spore formation. This is the most common form of asexual reproduction. And again, even organisms that can reproduce sexually as well, a lot of them will have asexual parts of their life cycle where spores are produced in this way. Okay, so sometimes a spore is just released from the mycelium itself. Okay, that spore lands, it starts to germinate, and we end up with a big mycelium. Or sometimes that spore is produced in a specialized structure, which is shown here inside of a sporangium, and then that spore gets released, and again, it's going to land and hopefully germinate and grow into a mycelium. Okay, now the spores in both of these cases are produced purely by mitosis. So there isn't, mating between two strains or anything like that. It's just a spore produced by mitosis. Again, either right from a hyphal strand or in a specialized structure, which we would call the sporangium. But in either case, it's asexual spore formation. Okay, so again, the perfect fungi reproduce both sexually as well as asexually. Imperfect fungi reproduce just asexually. For asexual reproduction, we could have budding, fragmentation, or asexual spore formation by mitosis. Sexual reproduction is different. So in sexual reproduction, you're going to have two mating types. Okay, this is kind of confusing. We don't call them male and female. Instead, we call them plus and minus strands. Okay, or we could say wild type and mutant. That's also used. In either case, we have two mating types, not male and female. We don't have egg and sperm produced. So it's very different from either animal or plant here. Now, sometimes sexual reproduction can be homophalic. You end up with a mycelium that actually self-fertilizes, kind of like self-fertilization in plants. Okay? Or heterophalic, and this is what we're going to do in lab. Phallic, hopefully, sexual reproduction. This is when you have two different mating types of two different mycelium that are growing in the same area that are genetically compatible with each other. Then you can get sexual reproduction. Okay, so you can have two mating types within the same mycelium. That's self-fertilization. That's called homophalic. Or heterophalic is two different myceliums with two different mating types that end up coming together. Okay, and I'm going to show you some pictures of this as well. There's three key events that are going to happen. It doesn't matter which species of fungi we're looking at. The life cycle will have the same events if it's a sexually reproducing fungus. So the first is called plasmogamy. Okay, so plasmo, cytoplasm. Okay, so what happens here is we have two haploid cells from two distinct mating types that fuse together. So we have two cells that fuse. Okay. We would call that for, well, let's wait there. Let's not call that yet. So we have two haploid cells, they fuse together. That's called plasmogamy. So pretty much their cytoplasms become one. And what we end up with here is the dikaryotic condition. Remember, we end up with, here's my hyphal strand, two nuclei within the same cell. We had two monokaryotic haploid cells, they fuse, and we get one dikaryotic cell, okay? Which means two nuclei in the same cell. The next event, it could happen really quickly, or there could be a little bit of a lag in between plasmogamy and the next step. The next step is called karyogamy. And so karyogamy is the fusion of those two nuclei. So now these two nuclei would become one. So the genetic material now from those two nuclei end up in one nucleus. Okay, so that cell right here is going to be called the zygote. That's the product of sexual reproduction. So we get plasmogamy is the fusion of cytoplasm. Karyogamy is the fusion of two haploid nuclei. So the zygote is going to be diploid. It has genetic information from both of those nuclei. And so it's 2N, or diploid, where each of these nuclei were 1N, or haploid. Okay, so now there's two alleles for every gene. Well, very quickly, that zygote is now going to undergo meiosis. And that's going to result in reduction of chromosome number back to the diploid, or the haploid state. Okay, so the diploid zygote undergoes meiosis, and meiosis is going to produce 4 haploid cells. And that might be it. Those cells develop into spores, or in some species, and the one that we're going to study in lab, this is true for, those 4 cells undergo a round of mitosis, creating now 8 spores, 8 haploid spores. And then those spores will be released. Okay, so for sexual reproduction, we've got plasmogamy, followed by karyogamy, followed by meiosis, and then sometimes followed by mitosis. Okay, let's take a break. We've covered a lot of ground here. So you're going to pause me, and you're going to go ahead and you're going to answer these questions on the Are You With Me? When you're finished, come on back. Okay, so let's make sure we're all together here. I got some questions for you. So the first question, which polysaccharide, or carbohydrate, is usually found in the cell walls of fungi? Is it starch, glycogen, chitin, or cellulose? All of these are polysaccharides. Which one is found in the cell walls of fungi? So hopefully you answered chitin. Okay, chitin is going to be found in the cell walls of fungi. The next question, which of these organelles is not found in a fungal cell? Chloroplasts, nucleus, mitochondria, or the Golgi? So hopefully you answered chloroplasts, right? Fungi are not autotrophic. They do not do photosynthesis. They do not contain chloroplasts. Next question. The wall dividing individual cells in a fungal filament, or a hyphal strand, we could say, is called is that divider called a thallus, a hypha, a mycelium, or a septum? So hopefully you answered a septum, right? Septa are going to be those structures, usually perforated, found in between individual cells in a monokaryotic or a dikaryotic fungal filament, right? Those would be absent in a coenocytic hyphal strand. Next question. The stage of the fungal sexual life cycle in which the nuclei of 2 different mating types fuses is it called mycology, plasmogamy, karyogamy, or sporogenesis? The fusion of the nuclei. Hopefully you answered karyogamy, right? Plasmogamy is the fusion of cytoplasm. Karyogamy is the fusion of the nuclei. And the result of that is going to be the formation of a zygote. So what happens next? Meiosis to produce 4 cells, and sometimes mitosis to produce 8, and those cells become spores. Okay. So we'll do a little bit of classification here, and then I'll tell you which groups we're going to focus on. So the common ancestor to all fungi on the planet would be at the base of this phylogenetic tree. You've probably seen these. You're going to see a couple of them this semester. Here, when we talk about animal evolution, we'll also look at a phylogenetic tree, or a lot of phylogenetic trees. So the common ancestor would be at the base. And so as we progress up, we're getting closer to present time. Okay, so we're going from the past forward to the present. Okay, and so at the divisions here, we've got the evolution of different phylums. Okay, and all of these phylums are present on the planet still today. Okay, so in evolutionary history, the first phylum to evolve on the planet, or division, were the chytrids. Okay, the Chytridiomycota. Your textbook does go into those, but I'm going to focus on the most abundant fungi on the planet and also the ones that we talk about in lab. We do not do the chytrids in lab, so I'm going to kind of skip that. Okay. The next group to evolve would be here, the zygomycetes or the zygomycota. We will be looking at those in both lecture and lab. And then the next to evolve here, the Glomeromycota. We're not going to look at those. Fungal names are kind of tough. The next group here to evolve are the Ascomycetes and the Ascomycota we are going to talk about. And the last group, or the most recent to evolve, are the Basidiomycota, which we are also going to talk about. Okay. About 95% of the fungi on our planet belong to the Asco and Basidiomycota, those two domains. Okay, so we're going to look at the Zygomycota, the Ascomycota, and the Basidiomycota. And again, you're going to look at those three divisions in lab this week as well. So let's look at the Zygomycota first. The Zygomycota is a small group of fungi that include the common bread mold that I'm sure most of you have seen. That bread mold's name is Rhizopus stolonifer, and you're going to actually see that in lab this week. So let me bring my picture up here. You can see right here, this is bread, and this part right here is the Rhizopus stolonifer, the Zygomycota. And if we were to look up close, this is what we would see. And this is neat. You can see a bunch of hyphal strands and stalk-like structures. And do you see those little black dots? Those black dots are all sporangia. Okay? Pretty neat. So inside of those sporangia, we have spores being produced. Okay? Most species of Zygomycota are going to be saprophytic, saprobes, meaning that they feed on dead, decaying matter. Okay. Few are parasitic. They are used commercially, some species, to produce hormones. Okay. If we were to take a look microscopically at their hyphal strands, they are coenocytic, so there's no septa dividing their cells, and they spend, again, most of their life cycle as haploid organisms. They will reproduce asexually, and so right here on this side, these are sporangia. Those are the products of asexual reproduction. If conditions are unfavorable, they will reproduce sexually. Okay, and in sexual reproduction, again, what's going to happen is we're going to increase variation, and sexual reproduction will result in two mating types coming together, forming a structure called the zygospore. And I'll show you a picture of that in the next slide. The zygospore has a really thick coat over it, which prevents it from drying out, desiccation. And when conditions become more favorable, when there's more moisture present in the environment, then that zygospore will kind of crack open, and it will release zygospores, and those can then germinate. Okay, so let's take a look here. So this is neat. Look at that. Pretty neat. Those are all sporangia. So here we have the stalk-like structure, and at the very top here is the actual sporangium itself, and inside of there spores are being produced. It's pretty neat. So this is the life cycle. This up here, this little tiny circle, represents asexual reproduction, and down here, the bottom part of this life cycle represents sexual reproduction. So let's start with a little small circle at the top. We'll start at number 1. Okay, so we have spores that are released. They land, they grow. They're going to produce this structure. Okay, we've got a lot of hyphal strands, and growing off of those hyphal strands are sporangia. Those sporangia will produce spores by mitosis. Those spores will get released, they'll land, and they start to grow again, and the life cycle just kind of goes around and around and around. Okay, now sometimes, again, if conditions favor this, we'll end up with sexual reproduction. So if we look right here, we've got 2 opposing but compatible mating types. We're going to call them the plus and minus mating types. If they, we've blown that up now, if they grow close to each other, then they confuse. We call them gametangia. There's no sperm and egg cell. They're not even really gametes. They're just 2 opposing but compatible mating types, a plus and a minus. They'll grow close enough together, and we get, here's that term, plasmogamy. They'll fuse. Okay, so here, this would be a dikaryotic nucleus. That's going to be followed by karyogamy, and we end up with a zygospore. Okay, inside of a zygosporangium. So that's the zygote. Okay, and then what's going to happen inside of here is meiosis, and meiosis will produce spores. Okay, so this will germinate, and spores will be released from this sporangium, and they are going to land and again grow by mitosis into a mycelium, where you can get sexual as well as asexual reproduction once again. Okay, so in lab this week, the first slide that you're going to look at is going to be this right here at the top. It's going to be a collection of hyphal strands with stem-like and root-like structures, with sporangiophores with sporangia that are producing spores. You'll see a collection, kind of a mass of those structures on that first slide, or I should say in that first picture. And then the second slide, you're going to see this. Kind of looks like 2 ice cream cones coming at each other, with the ice cream in the middle kind of being squashed. Okay, and that's the product, again, of sexual reproduction. Okay, so that was the zygomycota. Okay, again, our example in lab is going to be Rhizopus. You're going to be looking at 2 slides of Rhizopus. And then I think, one of the unknowns will belong to that division as well. So it will be a zygomycota. 2 of the unknowns will be ascomycota, and you're going to see examples of ascomycota as well before you get to the unknowns. So the ascomycota are also called the sac fungi, and that is because of a structure that they produce called the ascus. And so I'll kind of draw that for you. Ooh, maybe. The structure looks like this, and it's got 8. 1, 2, 3, 4. I can't do it. 1, 2. Drawing on here is hard. 3. Well, whatever. It's got 8 spores. I'll show you on the next slide. So the sac-like structure is called an ascus, and those 8 spores that are going to be produced inside of there are called ascospores. And the ascus is produced by sexual reproduction. Okay. Some are used commercially, so baking and brewing yeast belong to this group. These guys are unicellular though, so they don't produce asci with ascospores. A little bit different. You may have heard of truffles and morels. Those are edible mushrooms. And then also you may have heard of penicillium. That's an Ascomycota, a fungus that produces antibiotics, penicillin. Okay, there are some Ascomycota that are plant or animal parasites. So, for example, fungal pneumonia is caused by species of this group. If we look at the hyphal strands of members of this group, they are separated by septa, so they are monokaryotic. They will reproduce both sexually as well as asexually, some, not all. In asexual reproduction, we have structures called conidia that produce conidiospores. And sexual reproduction, that's where we get this. The ascus, which produces ascospores, which will be held inside of a structure called the ascocarp, or the fruiting body. Now again, in lab this week, you're going to look at 2 examples of this, and 2 of the unknowns will belong to this division as well. And then next week in lab, when we do the experiment with Sordaria, Sordaria formicola belongs to this division as well. So it's an Ascomycota. It will produce an ascocarp with asci that contain ascospores. Okay, so you want to get really familiar with this life cycle. So it's on the next slide. Let's look at that. Okay. So I pulled off a couple of parts of this life cycle and found some cool images. So we'll get to these images here on the left in just a second. Let's take a look at the life cycle here on the right. So let's start off at number 1. Okay, so we're going to start right here. Right here. Okay, so we've got hyphal strands that are growing, and we've got a plus and minus mating type. So actually, let's back up a little bit to here. So we have mating type plus and mating type minus. Okay, and so what you can see here is the plus mating type, we're representing the nuclei as red, and in the negative mating type, we're representing the nuclei with black, which means they're the same species, but they're different mating types. And therefore, their genetics are different. They're compatible. They have the same genes, but they may contain different alleles for those genes. So again, sexual reproduction would be the mixing of those genes, which would result in more variation. So you can see that we've got spores there that have started to grow by mitosis to produce the beginning of these hyphal strands. Okay, notice the hyphal strands. Can you see how they are monokaryotic? So again, we're looking at the plus and the minus right here. So here and here. Hopefully you notice that each one of the nuclei is in its own cell. So that's called a monokaryotic hyphal strand. So these continue to grow. And let's skip over to here. Okay. And at some point, they may produce structures that are going to be involved in sexual reproduction. And so what may happen is the positive mating type may start to grow over and fuse with the negative mating type, and that's called plasmogamy. And there we have this fusion. And when that fusion occurs, you're going to get this mixing of nuclei, or a migration of nuclei. That's called plasmogamy. So now we've got nuclei from both the plus and minus mating types in the same cell. So again, the cytoplasms have fused. That's plasmogamy. And then in some species, what happens next is we start to divide those cells up into dikaryotic cells. And so you can see here, we have a nucleus. In each of these cells, we have a nucleus from the positive and negative mating strand. And so now we have the dikaryotic condition. We have separate cells. Each cell has 2 nuclei from the 2 different mating strands. Okay? This structure right here will grow into the ascocarp. This is the fruiting body. Okay? And inside of that ascocarp, which is the cup-like structure, okay, you have cells in here that are dikaryotic. And what might happen is these 2 nuclei, the 2 haploid nuclei, 1 plus 1 minus, will fuse to produce a diploid zygote. Okay, so that's happening on that inner rim of that cup. You're getting the fusion of those two nuclei. Again, that is called karyogamy. So we had plasmogamy, we had the dikaryotic condition, now we have karyogamy, and now we have a diploid zygote. All of this is happening right here inside the ascocarp, the fruiting body. What will happen next is that zygote is going to immediately undergo meiosis. So we have meiosis 1, then meiosis 2. We have 4 cells now that are all haploid. And then each of those 4 cells undergo a round of mitosis to give us 8 haploid ascospores. And they're going to be inside of this sac-like structure. Let's go over here to this figure. That's what this is showing. So this is the inner rim of the ascocarp blown up. And you can see each one of these sac-like structures is called an ascus. And you can see that each of them should have 8 ascospores. Okay, we're going to be looking at these structures 2 weeks from now in lab, and we're going to actually be asking and counting the color pattern of the spores inside of there. Okay, so for the species that we're looking at, spores can be black, gray, tan, or clear. And it's going to be different. There's actually 2 experiments in this one. But you're going to be counting, or I'm going to be counting and giving you data for how many asci have, a 4-to-4 ratio or a 2-to-2 to 2-to-2 ratio. So we're going to be looking at color and pattern inside of these asci. Okay. When conditions are favorable and those spores have matured, they're going to be flung from the ascocarp. So this will open right up and they get flung out. Where they land, they're going to start to germinate and grow. And again, the cycle continues. Let's look at the asexual reproduction really quick. So again, you've got a plus and minus mating strand. For asexual reproduction, there's no plasmogamy. Okay, instead what happens is, in this example, we're showing the minus mating type. It will grow structures called conidiophore. And at the tips of those conidiophore, we get spores produced, which are called conidia, by mitosis. Those spores will be released, and again, they'll germinate and create a new mycelium. This figure right here, how cool is that? That is showing a transmission electron micrograph of a conidiophore with conidiospores at the tips. And actually, in lab this week, you're going to see the same exact picture of the slide. And the species that we're looking at, I'm not sure the species, but the genus is Penicillium. Okay? So you'll be looking at both asexual as well as sexual reproductive structures in the ascomycota. And then again, remember that in lab in a couple weeks when we look at the Sordaria, it is also an ascomycota. And hopefully we'll just be looking at sexual reproductive structures. That's what we're going to be concerned with. Okay, one more. So we did the zygomycota, the bread molds. We did the ascomycota, the sac fungi. And the last type here, the basidiomycota, which you're probably very familiar with as well, maybe most familiar with. These are the club fungi. So these guys produce a club-shaped fruiting body called the basidia. And you may have seen these, again, when I'm out hiking, I can't help myself. These are called bracket fungi or shelf fungi. You'll oftentimes see these on downed trees, okay? Or you may have seen something like this, right? This is a common mushroom. On the underside there, there's those gills. Okay, on the underside of these as well, there's gills. You may have seen smuts, rusts, puffballs. Okay. So again, they're called the club fungi. And the reason why they're called the club fungi is because on their gills, what we're going to find, and you're going to see these in lab as well, and on the next slide I'll show you too. On the gills, there's going to be these little club-shaped structures that are going to produce spores. Spores are usually have like this teardrop look to them. So this would be the basidia, the club-shaped structure. And these are the basidiospores, all 4 of those. Okay. Some of these guys are edible, but some are toxic, and many are pathogenic. Okay, so let's look at the life cycle. And again, you're going to see, I think, one example, and then one of the unknowns is a Basidiomycota as well in lab. Okay, so here we go. Let's start at number 1. So you got spores produced. You've got spores produced. Okay, and those spores, we've got 2 different mating types, right? Red and black, or plus and minus. Those spores are going to start to grow by mitosis. They grow into hyphal strands, which eventually develop into a mycelium. And then somewhere, if they grow close enough together, you will get the fusion of 2 cells, which is called plasmogamy. And then those cells will be dikaryotic, so there's going to be 2 nuclei in 1 cell, and those can grow into the basidiocarp. So the basidiocarp would be composed of dikaryotic cells. And then on that basidiocarp on the underside, we will have gills. And on those gills, right, here's a dikaryotic cell on those gills, we will end up with those cells, actually those, the nuclei in those cells will fuse, so that's karyogamy. And so we will end up with a zygote. And again, that's going to happen all across the gills. That zygote will then undergo, well, the zygote is the only diploid cell here. It will immediately undergo meiosis. Okay, so we have meiosis 1 followed by meiosis 2. So we end up with 4 spores being produced. Those spores are going to kind of migrate to the edge of the cell. And then eventually they'll be released from the tip there. Those are called basidiospores. So this club-like structure here would be on the gills, and it's called the basidia. Okay, those spores will then be released from the gills, and they'll be carried by wind, water, insects, animals, and wherever they land, hopefully they'll germinate, and we'll end up with another mycelium, which can then grow into a basidiocarp if sexual reproduction is happening. Okay. Okay, so your job now, let me move this, is to complete this next Are You With Me? You're going to just compare the life cycles of the 3 most abundant fungal divisions. And when you're finished, come back. I've got a couple questions for you. Okay, so the first question, members of which phylum produce a club-shaped structure that contains spores? Would that be the Chytridiomycota, the Basidiomycota, the Glomeromycota, or the Ascomycota? So the club-shaped structure. That would be the Basidiomycota. Remember that club-shaped structure is called the basidia, and it produces basidiospores. The next question, members of this phylum produce sac-like structures that contain spores. Is it the Ascomycota, the Basidiomycota, the Zygomycota, or the Chytridiomycota? So sac-like structures, the sac fungi, which are called the Ascomycota. Okay, I'm going to pause the video here, and I'll pick up on part 2.